Okay, welcome all, and thank you for joining today's AWRI webinar. My name is Michael Downey, and in this session we take a look at a Tasmanian Institute of Agriculture project investigating how Pinot Noir wine characteristics can be used to prove provenance and help industry better market their brand and product. But before we make a start, a couple of quick reminders for anyone that's new to AWRI webinars. To provide a comment or to ask a question, please open the Q&A section of the Zoom toolbar, type in your question and click to send it through. We will be holding a dedicated Q&A at the end of today's presentation, but please feel free to send through your questions at any stage. Also, a quick reminder, this webinar is being recorded and it will be available to view later this afternoon via the AWRI's YouTube channel. For those of you that have just joined, welcome. Today's webinar topic is Provenance of Pinot Noir, Drivers of Regionality. And joining us to discuss results from this project, we have Dr. Fiona Kerslake and colleague Dr. Rocco Longo. Fiona is Head of Horticulture at the Tasmanian Institute of Agriculture. And following completion of her PhD at the University of Tasmania, her research has focused on Pinot Noir and sparkling wine viticulture, developing capacity in wine analytical chemistry, and investigating drivers of sparkling wine composition. Rocco is a junior research fellow at the Tasmanian Institute of Agriculture and completed a PhD in wine chemistry at Charles Sturt University. Part of this work involved the development of winemaking practices for production of lower alcohol wines. And Rocco has expertise in sensory evaluation of wines and methodologies for measurement of aroma compounds. So Fiona and Rocco, it's great to have you both here to share results from this project. And Fiona, if you're ready to make a start, I'll hand it over to you to get us underway.
Great, thanks very much, Michael. So this is a project which was funded as a result of the Wine Australia Open Funding call back in 2016. So we've been working on the project since then, and it's really pleasing to get towards the end of the project. So it's concluding at the end of this year, and we'll be really pleased to deliver the final report to Wine Australia, which will be available on their website next year. So just like to introduce some of our research team. So it's been quite a large collaboration. So if you could just go to the next slide, please, Rocco. At UTAS, we've had a number of people involved, including plant physiologists, soil scientists, and also contribution from our business school for value chain analysis. We've also collaborated with Bob Danbergs at WineTQ and the AWRI. And also we've had initial design input from producers into how this project should look. But we're also incredibly grateful to the large number of Australian Pinot Noir producers who have given us an invaluable contribution towards this project, as you will see moving forward, and not just by providing their wines. And also this research was supported by funding from Wine Australia. And as we all know, Wine Australia invests in and manages research, development and extension on behalf of Australia's grape growers and winemakers and the Australian government. And this was also supported by the University of Tasmania. So next slide, please, Rocco. So Pinot Noir in Australia has quite a long history. It's only in 2019, Australia had slightly less hectares planted than New Zealand. In New Zealand, it's the top red variety crushed. However, in Australia, it only comprises around 6% of our total crush. But it's an important share of the domestic market and also very important in terms not only of volume, but in value. So the value of the Australian Pinot Noir market is quite high. In Tasmania, we have, thought I'd include this just for some context for vintage this year. Although yields were down this year and there was only around 5,500 tonnes crushed, the quality was quite high, but just thought it would be of interest to show that where our split at the moment is. So around 40% of our Pinot Noir crush at the moment is going to sparkling with the balance to our table Pinot, which we've focused on in this project. So looking at where Pinot Noir is grown across Australia, there's very many regions. So starting off in South Australia, you know, some of the key regions, the Adelaide Hills, the Eden Valley and the Fleurieu. Then as we move across to New South Wales with Henty and Orange, and also including Canberra district in the ACT. In Victoria, of course, we have a number of regions, including the Macedon Ranges, Geelong, the Mornington Peninsula, the Yarra Valley and Gippsland. Tasmania has a number of sub-regions, but some of the key Pinot Noir growing regions are our Tamar Valley Pipers Rivers region, Derwent Valley, Huon Valley, the Coal River Valley, and also the East Coast. But also important to remember that there's a great Pinot Noirs that are coming out of the Great Southern region over in Western Australia as well, which I was very fortunate to have the advantage of being able to visit a couple of years ago. And it definitely reminded me of some of the areas in Tasmania. So although Pinot Noir is usually described as a light-bodied red wine, frequent drinkers would know that despite this light colour and the lower anthocyanin and tannin levels, there's a really wide variety of wine styles which can be produced from the grapes. Some of this is a result of terroir or regionality and the area in which the grapes are grown. But then there are a number of vineyard and winemaking practices that the grapes respond to. So we were hoping to take a bit of a closer look at some of those aspects in this project. We also know that the aroma of Pinot Noir is quite complex. It would appear that we are yet to discover all of the contributing compounds. So this leaves sort of an area of intrigue that still surrounds Pinot Noir. But we've also just like to highlight that we have written a review of the literature to date in collaboration with the Brock University in Canada. And we've got the reference there for people if they would like to look that one up. But there's around sort of 25 to 30 volatile compounds which we've focused in on in this project. So as a brief overview of how we structured this project, we put it into 3 different wine trials. So we started quite broadly looking at a wide set of Australian Pinot Noirs with around 102 commercial wines from the 2015 and 2016 vintages. And accompanying that, we also had a survey which covered a number of different site characteristics, viticulture and winemaking practices. Then we narrowed the set down to around 80, and we included some aromatic composition analysis to see if we could also draw that out from the set of Australian wines. But then we also included some international Pinot Noirs into that set. So some from benchmark regions and some from newer and emerging regions. And then we narrowed it down to the pointy end at the beginning of this year, and we added a layer of sensory analysis over the top and with a smaller set of Australian Pinot Noirs with a tasting that we conducted here in Tasmania in collaboration with Wes Pearson from the AWRI. So I will just touch briefly on the survey and how some of the findings relate to the broader analysis of the 2015-16 wines. Then Rocco's going to take you on a bit more of an in-depth analysis of the last 2 trials, and particularly the last trial, because that was only completed this year, and that will build on the knowledge of people who perhaps may have seen Rocco and I present at the AWRI Pinot Noir Symposium last year. So by way of understanding what we covered in the Pinot Noir wine survey, there were around 170 questions in that survey. So when I say that our collaborating participants gave us a lot of time and effort, it really was a big commitment on their behalf. It was difficult to answer all of the questions for some of the participants, which is completely understandable. So it did limit somewhat the size of the data set, but also it's given us a really rich starting point from depth of information behind each of the wines to marry up with the compositional analysis. So although the survey was very much focused in on regionality, it soon became quite clear that we were also going to be gathering some information which could help us understand how you drive towards certain Pinot Noir wine styles. And we hope that that information as well as the regionality information is going to be useful to the Pinot Noir producers. The site and climate analysis portion of this is ongoing. We soon found that the information comes through from a range of different sources, and therefore standardised categorisation is quite challenging. So we have our soil scientist who's still working on scraping the data that he needs from the national databases so that we've got that standardised categorisation to work with there. Some of the survey outcomes that we had, this is just a few examples of the data output. So this is the kind of information which we're going to include in that final report that I mentioned, which will be posted on the Wine Australia website next year. But when we've completed surveys like this in the past in Tasmania, what we found is growers have found it useful for comparison to their own current plantings, or perhaps for making decisions on future plantings by understanding what others have chosen to do in the past. So we will, it won't look exactly like this, but we will put that into the final report so that that can help inform sort of current and future practices for Pinot Noir producers. And then what we were hoping from the survey data is that we would find some really clear, drivers of the wine composition that we were looking for. So the plots that we have here, this is relating the 2015 wine composition data for colour and phenolics to those survey questions. And it's a bit challenging to see in this plot, but what we didn't see was very clear clustering according to one single winemaking practice or vineyard practice. So I know many producers probably won't be very surprised by this finding, and that's due to the complex interplay between vineyard and winemaking, which variety like Pinot only makes even more challenging. However, in discussions with our soil scientists, some of the early findings that we're looking at in that area, we're very hopeful that we will see some of those drivers come out, and we'll gain some further insights once we've been able to complete that data set analysis. So that's a watch this space at this point. So I'm going to hand over to Dr. Rocco Longo now, who we are very fortunate came down to us at TIA from CSU once he completed his PhD. And he is our postdoc who's been working on this project team. So I'll hand over to Rocco now.
Thanks, Fiona, for your introduction. Hello, everyone. I'm going today to talk about two experiments that we carry out here at TIA over the last 2 years. The first one deals with a chemical comparison between single vineyards commercial Pinot Noir wines from Australia and non-Australian regions. Just to let you know that the manuscript associated with this study is currently in preparation. The subset of 80 samples used in this work included 40 Pinot Noir wines from 2015 and 2016 for a total of 80 wines. We selected these wines from 5 Australian regions: the Yarra Valley, the Mornington Peninsula in Victoria, Adelaide Hills in South Australia, Northern and Southern Tasmania. We also select three wines from 5 non-Australian regions, which were Willamette Valley in Oregon, Central Otago and Marlborough in New Zealand, Niagara Peninsula in Ontario, Canada. The same wines were selected from both vintages, and the 2015 and 2016 samples were analyzed 12 months apart, so the bottle aging effect were comparable. The experiment included analysis of the main color and phenolic attributes, and also of about 30 volatile compounds which are relevant to the aroma of Pinot Noir wines. This slide is about the first results that we had from this experiment. This is a principal component analysis of the volatile analysis results. On your left, you go Figure A. Figure A shows you the score, so basically the samples, while Figure B shows the loadings, which are the volatile compounds. From Figure B, we can see that most of the volatile compounds are actually in the upper right quadrant. These compounds here are mostly associated with fruity and floral characters, and appear to relate to the wines in the upper quadrant of figure A, in particular with the wines from northern Tasmania. You see this blue label NT, which means Northern Tasmania 2015 and 2016 year. As you can see from figure A, although a strong vintage effect is evident, as you can see from these two ellipses, red and blue, some samples were clustered by their region, such as Northern Tasmania, Ontario, and also the Mornington Peninsula wine, which is this sort of dark green. This is again a principal component analysis of the color and phenolic results. Figure A shows the samples, while figure B shows the chemicals variables. From figure B, you can see that most of the color attributes, color parameters, are in the right quadrants of the B plot. And these are mostly associated with the wines sit on this side. Specifically with, I can tell you that relate probably to these wines here, which are the wines from Oregon. As you can see from this slide, from this figure, you can also see that some samples, such as Ontario, this purple color label samples here, and Oregon and southern Tasmania. So basically, these samples here are clustering regardless of the vintage. So you can see there is a very nice cluster of 2015 and 2016. The second experiment which I will talk about today is on the sensory and chemical characterization of Pinot Noir wines from 5 Australian regions. This study is available online and is free for you to go and have a look if you're interested. For this experiment, we selected 3 Pinot Noir wines from 5 Australian regions. These regions were Adelaide Hills, Yarra Valley, Mornington Peninsula, northern and southern Tasmania. All samples were single vineyards. They were commercial wines. What we did was analyzing the wines according to the previous outline method, with the difference that we quantified a few extra color and phenolic attributes. And also we look at the sensory profile. The first results here that show this slide is a PCA of the color and phenolic parameters. Figure A again show you the samples, and figure B is the loadings plot show you these chemical attributes, which actually in this analysis here we just included those variables which were significantly different according to the ANWA. As you can see from this figure, southern Tasmania and Yarra Valley samples were found on the left quadrants of the B plot. And we can say that they are associated with total anthocyanins. On the other end, you can see the samples from Adelaide Hills, these red color labels, and Mornington Peninsula sit on the right quadrants, and that associate more to hue, chemical age 1, chemical age 2, non-bleachment pigment. This attributes. You describe the progressive change in a wine from deep purple red towards red brick and then brown color. And also all these attributes like chemical age index, they just describe how fast a wine is aging. Basically, the higher the value is, the older the wine will look like compared to the other sample that we see in this study. A different case is these wines from the northern Tasmania that we didn't see. They sit in the middle of the plot. It's quite challenging to interpret this result. Look like they sit somewhere in the middle. So basically, this result just suggests that Adelaide Hills and Mornington Peninsula seems to age quicker, while southern Tasmania and Yarra Valley have a more purple color. This PCA is on the results from the volatile analysis. Again, you can see the sample in figure A and the volatile compounds on figure B. This PCA includes only the significant attributes. We did that just to cut the noise from all the other volatile compounds, which we didn't see to change significantly across these regions. From figure A, you can see the northern Tasmania and Mornington Peninsula are found on the left quadrants. So basically there are similarity in terms of aroma composition. And these were associated more with ethyl-2-methylbutanoate and ethyl-2-methylpropanoate. These compounds are yeast metabolism byproducts. The first one is associated with apple aroma character, and the second one, ethyl-2-methylpropanoate, it reminds more sweet, fruity aroma. On the other end, you see these wines from Yarra Valley and southern Tasmania. You can also pick 2 or 3 wines from Adelaide Hills. This wine appear to relate to this compound, to these variables here on the right quadrant. Ecanoic acid has more cheesy character, while ethyl-decanoate is more fruity, grape-related aroma. The sensory analysis. So, we analyzed the wines for their sensory profile using a panel of wine professionals. Most of them were winemakers who generally charge at wine shop. We use a pivot profile analysis where descriptors are generated by the panel and then they are compared against the pivot, which in our case was a multi-regional unknocked Pinot Noir wines. As an example, when you practice this technique, you test the wine here on the right, and then you compare this wine with this one on the left. So, as an example, if the wine you're testing here has a more brown color than the pivot, then you need to write this descriptor on the right column. If the wine is less astringent and hot than the pivot, then you need to write this descriptor on the left column of the table. This slide is just a way to explain to you the principle of the pivot profile analysis. But in reality, we just use tablets and we did this just using a touch screen. So this is the first results that we had from the pivot profile. This is a frequency table made from the scores of the pivot profile analysis. This is a visual way to look at the tables and see what wines were scored high, low, for what attributes. The blue color means that wine was scored low for that attribute. And the gradient to the red is increasing, with a dark red the highest. As an example, H1, which means Adelaide Hills Wine 1, and Adelaide Hills 2, Wine 2, where both score higher for these attributes. Brown 10 times. This is the way you should interpret this table. On the results that we had from the pivot profile analysis, we also carry out a correspondence analysis. So this is a different plot than the one I showed you before. From the correspondence analysis of the sensory results, some regional effects are apparent. The Mornington Peninsula samples were associated more with red fruits and acidic. Adelaide Hills, all Adelaide Hills, including a few samples from Yarra Valley in southern Tasmania, a wine from southern Tasmania, were associated with brown color, complexity, and soft palate. They are also associated with floral and oaky aromas. An interesting thing to see in this plot is actually look at the samples from northern Tasmania, this NT blue, and also this one from southern Tasmania. As you can see, they spread out across the plot. We think that this is possible due to the fact that northern and southern Tasmania are bigger regions. For example, northern Tasmania include both Piper River and Tamar Valley, which and possibly this condition may dilute true regional effect on the sensory characteristic of the wines. So now I will end the presentation back to Fiona. She can discuss about some of the analysis that we've done on the value chain.
Thanks, Rocco. Yes, we do have another component to the project, as we mentioned earlier, with our School of Business and Economics. So Dr. Gemma Lewis has been working on value chain analysis, and she included some qualitative questions in the survey that we did online to underpin the wines. But she also has been conducting interviews with some of the Pinot Noir producers who indicated their willingness to participate in those interviews. So the main questions that she's been asking are around those two, which you can see there on your screen at the moment. And we don't really have time to go into great depth of those at the moment, but she will be including, of course, that in our final report. And there is also a paper there, which they published last year. And so in summary, I'd just like to say that, you know, we, sorry, just lost my place there for a sec. Could you just go to the next slide, please, Rocco? Thank you. So as I mentioned, the soil and climate contributions will be completed by the end of the year. We didn't find one single vineyard or winemaking driver of composition. Rather, it's looking like, you know, the complex interactions that we get and which we value in our Pinot Noir in Australia. What we saw across the 3 different vintages that we were looking at was that the volatile compositions of the wines that we looked at were more influenced by the vintage than they were by the region. However, we were seeing some signature colour and phenolic profiles coming through from the regions, which had a greater effect as compared to the vintages. And as Rocco was just describing there, we have some regions which have sort of quite distinct sensory profiles and distinct similarities between the wines from those regions. However, there are other regions which appear to have a blend of the different regions. So is that related to the size of the region and, you know, the topographical diversity and the range of microclimates? And I know in Tasmania, in one single region, you can get a huge variety of soils just within that region as well. That is quite likely. And then we also have the confounding influence of the number of Pinot Noir clones that we have, and then the performance of each of those clones at different sites and locations and soil types, etc. So it is quite a complex variety to work with, but I think as everyone knows who drinks it, it's also very rewarding. So further steps which we would, you know, ideally like to be able to take in this project is to source single clone fruit from a range of different regions and then use standardized winemaking to eliminate some of the potential winemaking influences that are coming through there. And also for the sensory analysis, we would quite like to take a look at the different, at a larger sample size. So thank you very much everyone for your attention today. It looks like we've got some questions coming through there, which I think Michael is going to shepherd us through now. So thank you very much. Our email addresses are there if you would also like to email us after the session.
Okay, fantastic. Thanks very much, Fiona and Rocco, for walking us through the results of those trials. As Fiona's already mentioned, we're going to roll from here straight into a Q&A. So Fiona and Rocco will stick around for a little while and answer any questions that you may have. So please do make use of this opportunity to ask a question. Please open the Q&A button in your Zoom toolbar, type your question and click to send it through. We do already have a couple of questions, which is great. So the first one is from Adrian. Given that Pinot is very subject to vintage conditions and yield, what variation between regions for each vintage and what correlations with ripening conditions and yield potentially did you see? Do these variations help explain regional differences?
There's definitely strong contribution from climate within each region, well, weather within each season within each region. And the yields were quite interesting. So there were some similarities amongst yields within regions, sorry. So that was quite interesting to see come through. But definitely the weather influence is a very strong one. So 2015, 2016 and 2018 were the 3 vintages that we used. But, you know, from a Tasmanian perspective, it would have been really interesting to have then had the comparison with 2020, which was so different again. And I guess we, you know, in the cool climate areas, we're very used to dealing with that variability, and we actually get to the point where we value it because it gives us some of that differentiation in our wines. But yeah, it definitely does have a big impact, and we've got further work to do on drawing out those correlations.
Okay, thanks Fiona, and thanks Adrian for your question there. Another question here that's been directed to you, Fiona. How do you know that the number of wines included in the sensory study are representative of the region and capture the different terroirs?
Oh, Marcus, we would love to do every wine in every region. That absolutely would be the ideal. But this was just a logistical decision that we had to do to drill it down as we added the additional layers of analysis over this project. And so that was actually a difficult choice, but they were the wines that we chose to go with for those regions. And they were based on, the selection was based on trying to get some diversity within the region, but then also part of it was in relation to those who had participated in our survey previously, and we're going to be able to provide some of the data to underpin the wines which were selected.
Okay, thank you, Fiona. Also got a question here about whether are there compounds associated with black cherry fruit characters?
That's a Rocco question.
Yeah, over to you, Rocco.
There will be some compounds among these that we have analyzed, specifically esters, that we can assume give this black cherry character. Those ethyl esters, we measure that, and to answer to your specific question, I just scrolled down that publication in relation to that that study, and I want to give you the correct answer before.
That's okay. I've got Pete's email address. We can email you the answer to that.
Yeah, look, if you want to go specific to the black cherry character, probably I need to come back to you. But definitely we didn't, given the analysis structure of the pivot profile analysis, we couldn't really draw this conclusion because of the nature of the pivot profile analysis. You can't, it will be very challenging then to try to build up a relationship between that aroma that eventually was generated by the panelists and the set of volatile compounds.
And if that characteristic specifically was used, Pete, as an individual characteristic generated by one of our panelists, it would have been included into the dark fruit aroma that we saw come through in the pivot profile. And it didn't really come through so much on the palette. It was sort of more in the aroma profile that we got more of those. We had red fruits in aroma and palette, but when it came to dark fruit, that really only came through generated as a descriptor for aroma, which was quite interesting.
Okay, thanks, Rocco. Thanks, Fiona.
Thank you.
Pete, if there are any further queries around that one, feel free to send through a follow-up question, or alternatively, Fiona and Rocco's contact details should be visible there on your screen now. So please feel free to follow up directly. We'll move on to the next question. Did you find that the violet's aroma in the wines, and does it have its own compound, or is it just subsumed into floral?
Yeah, basically the pivot profile, how the pivot profile works. You get a wine, you sniff the wine, and you, for example, can pick this violet. Then somebody else just fried floral. And then maybe somebody else fried rose. And so there is a combination of descriptors that are generated during this analysis. And then we need to draw down to one or two attributes. It is possible that these attributes, violet, was included into the floral descriptor that we have used.
So with the actual wine composition analysis, we did look at the norisopenoids, and so obviously with beta ironone being sort of the compound responsible for the violet characteristics, but also across all three, so the alpha ironone, the beta ironone, and the beta damascenone, we didn't actually find significant differences between the regions for either of those three compounds.
Okay. Thanks, Fiona, and thanks, Rocco. And thanks for your question there, Juliette. Another question here from Adrian. The University of Melbourne work is showing importance that the microbial biology on vine/grapes has on wine volatiles, showing huge difference between regions and between vineyards within regions. Did this project also look at vine microbial biology?
The short answer is no. It would have been great too, but yeah, unfortunately the short answer is no. It was beyond the scope.
Sure. Okay, question here from Scott. Was there any correlation of particular aromatics around ferment temperatures?
Rocco?
So if you refer to the last study, we didn't get this information from couldn't get this information from the winemakers. So I will assume that cool ferments, that wine produced by cooler fermentation will retain more of these volatile compounds, but that will be just an assumption.
We also, we have the opportunity actually, that's a very good question though, Scott, because we do have the opportunity to take a deeper dive into that with our 2015-2016 volatile composition. With the categories that we had were around sort of the lowest temperature, the temperature, and then the peak temperatures, but we actually could take a deeper look into that. And again, if you wanted to email one or both of us on that email address, if we have a chance to see if there are correlations there, then we can definitely follow that up with you.
Fantastic. Thanks, Fiona and Rocco, for that. And thank you, Scott, for your question. Again, if you do have follow-up questions, then feel free to contact directly via the details on the screen now. We don't have any further questions, Fiona and Rocco. Did you want to make any final comments before we look to wrap up?
I just want to thank all the producers who've given up their wine and their time in providing all the information that sort of underpins this. It's we're really grateful. We can't do it without you know, such a high level of collaboration, and we're very grateful.
Great, thank you, and thank you to Fiona and Rocco for yeah, coming in and sharing results from this project. Really appreciate you sharing some information around this. Thank you also to the audience who's logged in and joined in today's session. A reminder to everyone that you will receive a follow-up email with a link to the recording of this session. Also a reminder that Fiona and Rocco referred to a few papers in their presentation. I think a lot of those are available to request via the AWRI website, but you can also contact the AWRI library directly. Acknowledgements to Wine Australia for providing funding and support for AWRI webinars via the Extension project. Also just some information about the next AWRI webinar, which is on Thursday, the 29th of October. And will be presented by the AWRI and Yarra Valley Wine Growers Association, and takes a look at a range of topics related to vineyard viruses. If you haven't registered for that session and would like to do so, then please visit the AWRI website. That's all we have for today. Thank you again to Fiona and Rocco, and I look forward to seeing you all at the next AWRI webinar.